So let's move on to talking about what actually makes up our genomes. So our genomes are made up of genes and other genetic elements, either information that is used in some other manner related to the genes or simply structural elements for the genome. So just a quick review from the last lecture video. We have our karyotype here, and we have all of our chromosomes, human chromosomes. And within each chromosome, there is a linear pattern or series of genes. So all along these chromosomes, you have genes encoding various characteristics within an individual. So for example, on chromosome 9 here, we have the gene that codes for the ABO blood type. So let's talk about this notation real quick. 9 means chromosome 9, so we see chromosome 9 here. Q means the longer arm, and you can sort of see this arm down here is a little bit longer. Position 34, so that is basically a map number, a linear number that shows where that gene is along the chromosome. Now remember, a locus or loci is the plural, is the location of a specific gene on the chromosome. So this 9q34 gives us a number to identify that location on a chromosome. Now you can look at the total genetic map. That is the sort of the atlas of all the genes in a genome. And we saw that in the last lecture video, I believe, where we looked at the G staining of the karyotypes, and it had listed those different types of stains correspond to different locations. And there are other maps available. We have one in our genetics classroom that shows all the numbers and positions and even labels some of those. You can find those on the internet as well. If you just Google, say, chromosome 12 map, you'll find those. So not all of the genome has genetic coding information. Some of the genome consists of genes, and some of the genome consists of other types of DNA. So in the human genome, not all of our code actually codes for actual functioning genes. So when we look at a sequence, we can sort of think about what it does. If we have a particular location within the gene, is that location transcribed? That means, is it turned into messenger RNA or some other types of RNA? And we'll talk about RNA later. Is it transcribed? Does it actually produce a protein and therefore a phenotype? Or there's other functional reasons that you might have a particular bit of information. It might be a recognition site for something like beginning of replication, transcription, or even transcription factors. Transcription factors. So each genome is different, and within each genome, the exact percentage of the genome that encodes expressed proteins varies among organisms. Now, in general, bacteria and other prokaryotes have genomes that are almost fully functional. That means they have basically no extra DNA. Everything is coding or provides a particular purpose within the organism. In eukaryotes, though, we have a bit of a different story. So in eukaryotes, we often have parts of the genome called exons. These are expressed portions of the genome. That means we get a code for a protein and therefore a phenotype. And then we have other regions that are transcribed but not necessarily expressed. And these are our regions that are going to make other types of RNA. They're going to be involved in transcriptional control. And other non-protein expression functions. In humans, we see that actually only a very small percent, about 2% of our genome, is actually expressed and transcribed. The rest of it, the rest of the functional part, is non-transcribed. So we have a very small percentage of non-transcribed, a very small percentage of information that's actually expressed. But still, a lot of it is about 70% has a function within the genome. Now, you can also have regions, particularly in eukaryotes, not so often in bacteria, that is non-coding. So these non-coding regions may have some sort of structural function. For example, telomeres. They may be DNA that's no longer needed and has probably acquired some sort of mutation so that it's no longer functional, or it may just be unknown function. So it's not that it doesn't necessarily have a function, but we just might not know what that function is yet. So in general, more complex organisms have more protein coding genes. But the flip side of that is that they also make up a smaller percentage of the DNA. So your prokaryotes are your eukaryotes have more non-expressed sequences and to some extent more non-coding DNA. Humans have a lot more of this non-coding section. Recall from earlier in our Chapter 3 discussion that in general, more complex organisms have larger genomes, and so you are also going to have larger regions that are transcribed and are expressed than you would in smaller organisms. As part of genomes and part of the makeup of genomes, you can expect genetic change. Now, genetic change can happen in a variety of ways, and it all is going to affect the evolutionary history of an organism. But let's talk about some of the ways that genomes change. Now, keep in mind that not all changes have a functional consequence, and in fact, most of them don't. So let's talk about why that is. So if we have a single base pair change, and if the chance of that having a consequence for the organism is low, then where do we get this diversity that natural selection can act on? So in other words, organisms have mechanisms built in to limit the problems that arise due to changes in a genome. So if we limit these changes, how do we actually get the diversity that we need for natural selection? Well, it comes from many places. One is that we have many mutations that occur in the genome. Now, this can occur because of environmental influence. It can also occur when DNA replication takes place, and there are errors that happen during DNA replication, although that process corrects for errors as well. Also keep in mind that genomes are really big, so there are many, many places where changes can occur. And then, of course, we have many individuals, whether we're talking about humans or insects, there are many, many individuals. And then, of course, over evolutionary time, we can accumulate a lot of different information. So when you put all of these things together, many mutations at many locations among many individuals during a long period of time, you get sort of this accumulated effect of genetic diversity. So you basically accumulate variation on which natural selection can work. So even though most of the mutations don't actually have significant consequences, the ones that are affecting genetic diversity, the ones that do have a genetic consequence, are likely to cause phenotypic differences and therefore evolutionary change. And so when we have variation at the molecular level, we get change in phenotype and therefore change in populations. Even though those initial changes are unlikely to have much consequence. All right, so another way that we can limit the effects of mutation, the last slide was talking about how we actually accumulate mutations. We can also limit the number of mutations that occur. Now, some of this is limited in DNA replication. So DNA replication actually has proofreading mechanisms within it, so you can limit the number of single mutations within the DNA. But if a mutation does occur, there are certain ways that the genome sort of combats this effect of mutations. Now, one is redundancy. So redundancy is where you have one or more versions of the same information. So essentially, this means multiple copies of the same gene that are functional within the genome. So let's compare this to a road. We might have one road with one lane in each direction, leading between town 1 and town 2. So we might have a pathway that leads between phenotype 1 and phenotype 2. So maybe this is a metabolic pathway. Now, redundancy would be made by making a divided highway. So now we have more lanes. We have 2 lanes going in each direction. So even if one lane is blocked, or let's say we have a mutation that makes that lane dysfunctional, we still have another lane, thereby ensuring that that actual pathway can still proceed. So you can get redundancy in a couple of ways. One of them is something called degeneracy in the genetic code. Now, we'll come back to this in a lot more detail later, but basically this means that if you have an mRNA codon, remember that the messenger RNA is read in sets of 3 to produce the amino acid chain. So degeneracy means that in the genetic code, built into the genetic code, you might have a third base pair that differs. So the actual important part is the UA. So that UAG might encode for a specific amino acid, but UAC also codes for the same one, and UAA codes for the same one. So if you have a mutation that changes this codon from UAG to UAC, you're not actually going to change your amino acid. We can also have multiple members of the same gene family. So this is essentially duplicate copies, either identical or related, that do the same thing. And then on top of that, diploid organisms have 2 copies of every gene. Remember, one from mom and one from dad. So even if you have one dysfunctional copy, there's potentially the other functional copy in your other parent. Another way to combat the effects of mutation is something called robustness. So if we look at this image here, robustness gives us alternate routes to the same outcome. So down here between towns 3 and 5, this would be our example of redundancy. We have multiple lanes to get between towns 3 and 5. Same thing between towns 2 and 5. In fact, we even have more redundancy here. But on top of that, we also have robustness. So you can get from town 1 to town 5 in multiple ways. You can go from town 1 to town 2 and get to town 5. Or you can go from town 1 to 3 to 5. Or you can go from town 1 to 4 to 5. All of those pathways get you to the same end result. Now, genes can do that in the same way. You can have multiple genetic pathways or multiple genetic mechanisms to get you to the same expressed phenotype. So essentially, robustness is having multiple pathways to get you to the same expression pattern. So this might be multiple types of genes, or it might be a combination of both. You might have genes doing two related genes doing very similar or even identical parts of different pathways that are all going to get you to the same end. So let's look at what happens to the genome when genetic changes actually do occur. So let's look at how those changes actually affect the phenotypic or morphological differences within an organism. So at the lowest level, change is going to occur at the base pair sequence level. So when we compare different genomes, we can see differences in those sequences. So our top sequence here is looking at this region here, and this is the normal or the wild type. So wild type just means sort of the most common sequence or most common genotype or phenotype in a population. These are also sometimes called unaffected sequences, and they don't necessarily mean bad. That doesn't mean that the non-wild type, the non-normal, the affected are negative. It just simply means that we have different versions. Now, of course, they can refer to disease or other sorts of issues for an organism, but they are not always. So if we have a normal individual with a T here, there are different changes that we can see in a genome. So we can have a base substitution. So in this case, we have substituted our T for a C. Now, you might be thinking, wow, this is probably going to affect the amino acid sequence. And if you're not, I'm going to tell you that this is probably going to affect the amino acid sequence. Remember that our amino acids are read in triplet, so if you change one of them, you're very likely to change the code that you are using. So there are several ways that we can affect our sequence. One, we can have a neutral effect. This means no change occurs, and remember that has to do with degeneracy of the genetic code that I talked about just a minute ago, where you can have a change in that third base pair of the codon and it doesn't actually affect the amino acid sequence. It doesn't produce a changed amino acid. You can also have a missense. Now, this is a mutation that changes the amino acid in your sequence. So it changes the amino acid that is put into the polypeptide chain and therefore is probably going to at some level affect your protein structure. Now, depending on which amino acid is changed depends on how much structural change we actually cause. Then you can also have a nonsense. Now, a nonsense mutation essentially introduces a stop codon. In other words, it means that it terminates the sequence and your protein stops. Now, if you're missing a chunk of your protein, you are not going to have proper folding. You're not going to have an overall functional protein. And so nonsense mutations are in general the most detrimental. Now let's say that we add or remove a base. These are called indels for insertion or deletion. So if we have an indel, we are going to introduce something called a frame shift. So remember, we read in codons of 3. So in this case, we should have TCT, but when we delete a base, we actually code TCA instead, and then we shift our reading frame. So we shift the set of 3 that we're looking at. So here's our shift here. Now we can shift it backwards, so this would be a shift backwards due to a deletion, or we can shift it forward. So if we are looking at this set, we actually if we introduce, we can shift it the other way. So either way, we are changing the entire amino acid sequence past where we are producing our indel. Now, this is going to lead to a whole bunch of missense for sure. It's very possible that you are also going to introduce a stop codon at some point and therefore make it a missense as well. Insertions and deletions are probably going to have a big effect on your overall structure because you're changing everything after that indel, whereas a base substitution is likely to change only one amino acid, so it's less likely to change the overall function greatly, although it certainly can. There's a potential for it not being quite as bad. Now we can also insert something called a copy number variation. So a copy number variation is where you have a string of nucleotides that are copied and repeated in multiples. Now, this happens for a variety of reasons we'll talk about a little bit later. We're not going to get into how it happens. I just want you to be aware that it happens. So Huntington's disease, you might be familiar with this. Huntington's disease is a neurological disorder that causes deterioration of all sorts of things, neurological function and muscles and things like that. Huntington's disease occurs more commonly in men and over about 40. So this is a genetic disorder that you are born with, but doesn't appear until basically your middle age. And the higher copy number variations in Huntington's disease leads to more severe disorder. Now with this disease, within the gene that causes Huntington's disease, we actually all have a certain copy number variation, but once you hit a certain point, the disease begins to manifest, and the higher number of copy number variations you have, the more severe that disease is. All right, so we can have changes at the DNA sequence level, but we can also have changes in genome size or the number of chromosomes. So mutations are generally changes in the sequence, but we can also have other structural changes. You can have changes in the number of chromosomes. You can also have changes in the physical anatomy of specific chromosomes. So remember from earlier in this chapter, we said that genome size generally increases, but it rarely decreases. And these increases essentially come from duplications. So duplicating part or the entirety of the genome. So let's talk about some of these changes. The first is polyploidization. So you might recognize this ploid. Remember that ploid means number of chromosomes. Okay, poly means many. So this means many copies of chromosomes, or changing the number of chromosomes. So we are diploid, we have 2 copies of each type. Some organisms might have 4 copies of each type or 6 copies of each type. This is very common in plants. There's an example here on the right that shows some common ploidies in plants. So wheat is 6N equals 42. That means there are 42 total chromosomes, 6 copies of each type. And then we've got some weird ones down here. Boysenberry, 7N equals 49. Banana, 3N equals 27. When you have these odd numbers of chromosomes, that actually affects meiosis and cell division and reproduction. So the odd numbers tend to be things that are cultivated by hand. But a lot of our crops, you'll notice that all of these are things that we eat or use. All of these are crops, and polyploidization actually can have some specific effects on the plant itself. So organisms with more chromosomes tend to have particular qualities about them that are useful in crop plants. So there are two ways that polyploids come about. We're not going to spend too much time on them. I'm just going to briefly tell you what they are and how they work. In allopolyploidy, you have the combination of chromosomes for two different species. So you might have species A with a total of 6 chromosomes and species B with a total of 4 chromosomes. Now, in general, two different species can't breed, but if they do, and if they do it successfully, you are going to end up with a new individual that has 2n equals 10. So we have changed our number of chromosomes when we were able to successfully combine them. Now, keep in mind that polyploidy tends to happen more in plants than it does in animals. Now, when you change your number of chromosomes, you're going to affect who that individual can breed with. So this individual that is now 2n equals 10 is no longer going to be able to breed with either species A or species B. So you get a new species. You can also have autopolyploidy. So this is essentially self-polyploidy. This is when, due to some sort of an error in fertilization or in meiosis, you get one parent essentially doubling or ending up with an additional copy of their chromosomes. So a couple different options here are going from diploid to triploid, where you get one copy of each of the chromosomes you already have, or tetraploidy, where you get a copy of both chromosomes that you already have. The difference here is that allopolyploidy is a combination of two different species. Autopolyploidy is one parent ending up with duplicated genetic material and passing on that to the next generation. So again, we can also have changes in the number of copies of a particular sequence. Again, this is a copy number variation. We saw that a minute ago, and I've sort of already explained that. This is where you have a block of DNA sequence, usually about 1,000 kilobases or more, duplicated and repeated many times. Now, this is important because, one, it produces redundancy. If this happens to duplicate a whole gene, then you have different genes. The other effect that you have is this is not producing an entirely new gene, but it is probably going to affect gene function and therefore for protein function, because you're inserting new bits into that genome. Now, this might be advantageous or it might not be. So we have the potential for natural selection to work here. And of course, natural selection can work on those mutations and on your polyploid situations as well. All right. Another way that you can increase the size of your genome is by producing gene families via duplication. So a gene family is a set of genes within a genome that have similar sequence and therefore similar or highly related functions. Now, again, you can have the potential for redundancy here, which just means you have two different genes that are going to get you to the same pathway. But the cool thing about gene duplication is either you have redundancy or you have another copy or more than one copy that can actually acquire mutations and can acquire change. And so when we copy our genes within our genome, some of those duplicated genes can actually diverge and take on new, this can be novel or related functions. So we now have a sequence that evolution can actually act upon. And this is, if you go back to the Blackboard folder for Chapter 3, there is a book publisher video on something called Hox genes. So Hox genes are a set of developmental genes that can be traced for duplication and divergence and have been associated with a lot of different developmental stages within living organisms. So here we have showing how this might happen. We have an ancestral gene. That gene is duplicated. And it might be duplicated again, might not be, but either way, they are going to potentially diverge into other genes. So these are, the bottom line here are similar genes to the ancestral gene, but they all have different but related functions. And then they've also shown this gene here in black, which is one that has acquired mutation and is no longer functional. We call that a pseudogene. So a pseudogene is a gene that is a region that used to be a gene that doesn't work anymore. And then all of those genes that are in the related gene families are called paralogs. So gene A1A, A1B, and A2, those are all paralogs. All of these genes are paralogs. They are similar but related functions. All right, so here is an example of basically duplicated genes that provide a specific function in humans. Now, generally, within the same species, you do not have different numbers of genes because you have one of the things that we said was that in homologous chromosomes, you have a specific set of genes, and there might be variation of the version of the gene, but you're going to have all the same genes. But we have this really cool example of gene family paralogs in an enzyme called amylase. So amylase is a gene that breaks down starch. Humans have this, and ancestral populations that have come from regions where there are high starch foods such as potatoes or other high starch crops have individuals that tend to have more copies of the amylase gene. And individuals whose ancestral population had less starch in their diets tend to have a lower number of copies of this amylase gene. So this is kind of interesting because it's unique in that humans generally don't differ in their copy numbers of genes. They can differ in their copy number variations within sequences, but they generally do not vary by copy number of genes. But you can look at different populations. So we've got the high starch diet population and the low starch diet population. So this graph is showing what percentage of the population has this number of genes. So for amylase gene 4, individuals have multiple copies of gene 4 if they are in low starch regions. But some individuals have more copies, and those individuals that come from populations with high starch diets actually have more copies of those genes. So an increase in the number of paralogs likely provided some sort of nutritional advantage for those individuals who were in areas where they had access to high starch diets. This is going to mean they had an increase in fitness, but on the converse side of that, if you have a high number of amylase copies, but you're eating a low starch diet, you're not really going to have much of an advantage because those genes are not going to confer any more ability than anyone else in your population. So just sort of a summary to wrap us up here. If you have changes in the genome, you are very likely going to lead to changes in phenotype. Now these changes are going to come from genetic changes in your genome that cause changes in amino acid sequences. So this is going to affect protein function. We can also have changes in gene regulation. So this is going to change when, where, and how much protein is expressed. Variation in splicing, this is something that we will get to later. This is variation in piecing together your expressed sequences. So when you have variation in splicing, you can get variation in protein types in protein products from the same gene. Changes the genome can also lead to novel genes and novel functions. So this is when you get your duplication and divergence. And then you can also get changes involving RNA encoding genes. So these are things, these are changes that affect functional RNA molecules within a cell. So RNA molecules work to regulate gene sequences. They are also sometimes structural molecules that do a particular job within a cell. So of course, if you have changes in your genome, we talked about some of those: substitution, indels, copy number, gene families, ploidy, you are going to likely produce functional changes and therefore phenotypic changes as well.